Thanks, Tim. So it's a real honor for me, actually, to get this opportunity to give a talk in a symposium honoring Gobi and Khurana. 32 years ago, I was a freshly graduated organic chemist working for what at the time was, I think, the only company in the United States that was trying to make commodity chemicals out of agricultural biomass. And one day when I was reading the literature, I came across an issue of the Journal of Biological Chemistry that had 17 papers in it from Gobi and Khurana's lab describing the total synthesis of a tRNA synthetase gene and showing that it was active. And as an organic chemist, the total synthesis of an active gene had particular meaning to me and led me to think there really must be something to this recombinant DNA stuff. It's going to have all kinds of potential. And I actually wrote up a little white paper for the company I was working for and tried to convince them that they should try to engineer bacteria to convert biomass into these commodity chemicals. But that was received at the time like a lead balloon, and I probably smartly went to graduate school and learned about recombinant DNA and went on to have a really interesting 25-year career studying another subject that Gobi and Khurana made fundamental contributions to, transcriptional regulation, and in my case, particularly the structure function of RNA polymerase. But curiously, I'm sort of coming full circle, and I'm going to have the opportunity here to come back and describe something about the conversion of biomass. So about 2 years ago, we found out we were going to have the distinct pleasure of setting up in a university culture a research center devoted to trying to create bioenergy and biofuel molecules. And I'm going to take as a given that you understand why it's important for molecular biologists to do something about getting the country off foreign oil and producing renewable fuels. And I'm going to talk about work done by this group of researchers that's in the last year and a half been put together here, both with full-time staff by GLBRC and UW faculty and researchers that are contributing to the effort. And I want to back up a step from what you were hearing about from Jay and tell you a little bit about why this is such a difficult problem, what it is that has to happen to convert cellulosic material into biofuel, and then secondarily a little bit about some of our genome engineering efforts. So why is it so hard to make fuel from cellulose? And well, let me say first, there are sort of 3 steps in this process, and they involve taking the biomass and chemically pre-treating it to expose the cellulose, using enzymes to release the sugars, and then using microbes to convert those sugars into some fuel molecules. And the reason this is so difficult has to do with the structure of the lignocellulose, which nature has very deviously put together to make almost impervious to any kind of biological attack. So the sugars are tied up in these cellulose fibers that are packed in crystalline arrays, which means the enzymes can't get at them. They're wrapped in hemicellulose that has a wide variety of linkages, meaning that it takes a large number of enzymes to figure out how to break it down. And then those are further packaged up by lignin, which is actually stereochemically randomized in the process of synthesis, which means it's really impervious to any kind of biological attack. Enzymes can't degrade it. And there are really 2 inexpensive ways to try to get at it. You can treat it with acid, and that releases the C5 sugars, but it converts the lignin to phenolics that are toxic to most microbes. Or you can treat it with base, and that actually isomerizes the cellulose and redeposits the lignin to the surface of the biomass. So in the GLBRC, we're using a base process, ammonia fiber expansion, invented by a colleague, Bruce Dale at Michigan State, who's a key contributor to the GLBRC. And this slide from Bruce really illustrates the difference in sort of the ammonia pretreatment and the acid pretreatment. Particularly the acid pretreatment leads to both a soluble and an insoluble feedstock that then have to be fermented, versus this AFEX treated material that leads to a single stream that can then be enzymatically hydrolyzed and fermented. And so as microbiologists now, this is our new bottle of glucose. We have to figure out how to take this material and make it into something useful. And I think you can appreciate both from the single stream and sort of for aesthetic reasons why it's preferable to start with the AFEX treated material. So it's difficult to break this down, as I said, in part because you need a large number of different enzymes to break it down. The cellulose requires enzymes that degrade from both ends, release cellobiose subunits, and those have to be cleaved, and hemicellulose is equally complex. And there's sets of enzymes that I am not even representing on the substrates here that have to be used to break it down. So actually, the full suite of enzymes is something that is still not completely worked out. We know sort of a minimal set that does most of the job, but work is ongoing to figure out really what the true set is that would be needed. And you need not only a large variety of these, but you need them in very large quantities. So to break down starch, it takes milligrams per kilogram of biomass to break the glycosidic bond. And it's amazing what one simple change in stereochemistry does to this problem. Cellulose, which forms these crystalline arrays, now requires grams of enzyme per kilogram of biomass to convert it into sugars. And so that means it's very difficult to generate the amount of enzyme that's needed. It's an energetically costly process to do that. And to make it inexpensive and to make cellulosic biofuels a reality, something has to be done to make those enzymes inexpensive or make them vastly more efficient. So to just illustrate that problem, this is an economic calculation done by Bruce Dale's lab, just showing that if you could, in fact, create all of the enzymes needed using the same organisms that were converting the sugars into biofuels, which is called consolidated bioprocessing in the field of bioenergy, you could reduce the cost of producing ethanol almost by a factor of 2, coupled with some other processing advantages to doing that. But there's a real dilemma in this from the biological standpoint. That is, you need to get a lot of energy in order to make these enzymes, right? And that's done best aerobically, because you can make a lot of ATP aerobically. It takes about 1,500 ATP molecules to make a single protein. And yet, when you make biofuel, what you're trying to do is conserve all that energy. And that's actually best done anaerobically, where you can do fermentation and, for instance, make ethanol and conserve nearly all the energy. The cell only gets 2 ATPs out of this, and you can make fuel. And so how to resolve then this dilemma of a very energy-intensive process that you need for making enzymes with the need to conserve energy to make biofuels. And the way we're trying to approach this in GLBRC is a strategy of engineering microbes so that we could use the anaerobiosis actually as a regulatory switch, such that in the presence of air or oxygen, we could grow the microbes, produce the enzymes, and then upon withdrawing the air, switch the metabolism of the bugs to converting the sugars that are released into biofuel. And we're focused particularly on ethanol at the moment. You heard from Jay some of the arguments for why other fuels have advantage, and those are all true, and everything I'm telling you would be equally applicable were we trying to make some of these advanced biofuels. I think for the most part in GLBRC right now, we're focused on ethanol as a proxy for this. And we're doing these studies using both E. coli as a model organism, Saccharomyces, and a gram-positive lactobacillus. But what I'm going to tell you about for the rest of the talk today really is focused on the work just with E. coli. And the strategy there to try to convert E. coli to an organism that can efficiently make these enzymes and be able to break down the lignocellulose is to take advantage of sort of a two-step strategy. So first of all, to identify close relatives of E. coli that are already able to degrade AFEX-treated corn stover efficiently. And we're doing this in collaboration with Nicole Perna and Cameron Curry's group, focusing on close relatives of E. coli that grow on plants, enterobacterial phytopathogens, and other gamma proteobacteria that come out of leafcutter ant colonies, and then trying to find microbes that can break down the substrate. An interesting thing about these systems in gamma proteobacteria is that they're almost always use type 2 secretion systems to secrete the glycosyl-hydrolases. And while the native system in E. coli is actually transcriptionally silenced, represented here, the systems are almost always present as gene clusters, such as the one that's found in K12. And it's already been shown that you can transplant those into E. coli K12 and get them to be active. And so a good strategy then is to find a good bug, transplant in the secretion system, figure out the set of genes that need the bug uses to break down the AFEX-treated corn stover, transplant that in, and convert the bug into one that can break down the lignocellulose. So from these ant colonies that Cameron Curry has been working with, which are actually prodigious breakdown or prodigious degraders of biomass. They can actually turn over about 1/2 ton a year in these large ant colonies. This shows a picture of one being excavated here by people. You can see how big it is here. So they do it microbially, both because the ants are using it to grow fungus that they use for food, but also they use these microbes in sort of refuse dumps to break down the recalcitrant part of the biomass. So they're very good sources of bugs that are able to break down lignocellulose. And in fact, we already have large numbers of gamma proteobacterial candidates out of Cameron Curry's efforts, many of which now we're in the process of screening for their ability to break down AFEX-treated corn stover, and some of which are already out at the Joint Genome Institute being sequenced so that we can get these genes out of them. And just to show you one example of this, this is one of the attractive candidates we have in hand already, comparing what happens if you take E. coli, K12, or actually the strain that Lonnie Ingram has engineered to make ethanol, and try to grow it on this AFEX-treated corn stover compared to this gamma proteobacterial candidate, which is actually able to fairly efficiently convert all of the AFEX-treated material sitting on the bottom of the flask here with E. coli into biomass. And so we're conveniently, this happens to be one that's already been sequenced, so we're already engaged in the process of trying to transplant the secretion system and identify the genes that are responsible for this breakdown. So that's essentially what I want to tell you about the first part of this. And in the second part of the talk, I want to tell you about our efforts so far to do genome-scale engineering to really understand the anaerobiosis regulatory network as a way to execute this strategy I've described to you. And that recall that the idea is we have to understand what the oxygen regulatory mechanism is that controls gene expression in E. coli so that we can take advantage of it to program bacteria to carry out these processes. So this is, at a metabolic level, is a complex but relatively well understood problem in E. coli. And E. coli actually regulates the switch from growing aerobically, where it converts glucose into energy, actually by breaking it down, stripping off the electrons in the TCA cycle, and using efficient electron transport chains to drive this proton motive force and then make ATP using ATP synthase. As E. coli switches over to anaerobic growth, it first switches over to, in low oxygen tensions, higher efficiency electron transport enzymes. And then eventually, in the absence of oxygen, converting over to fermentive growth, breaking apart the TCA cycle into pathways that generate substrates for building up cells. It actually makes a large number of alternative electron acceptors in the membrane at that point, or enzymes for using alternative electron acceptors, even when they're not present. And in the absence of any electron acceptors, it simply carries out fermentation, converts the glucose. And actually in E. coli, as Jay mentioned, it tends to convert the glucose into either lactate or acetate. And the reason for that is that in order to get to ethanol, it needs 3 NADHs to get from pyruvate ethanol. And it only has one available per pyruvate in the fermentative pathway. As a result, it tends to make these acids. That problem was solved by Lonnie Ingram a number of years ago by putting in the pyruvate decarboxylase genes from Zymomonas mobilis, and they actually will take pyruvate and using a single ADH and decarboxylase pathway make ethanol. And so we have now recapitulated the sorts of strains that Lonnie has made. You can knock out the lactate dehydrogenase, acetate kinase, put in this pyruvate decarboxylase. It's actually put in coupled to the pyruvate formate lyase gene, which is something that's reduced heavily in the anaerobic environment. And these strains actually will make high levels of ethanol growing on glucose. So we're now in the process of characterizing these from a systems biology standpoint. And we've put together, to do that, a fairly large suite of different kinds of omic analyses and an experimental fermentation lab that's busy growing these strains. We've put together ways to look at transcriptomics, metabolomics, lipidomics, proteomics, ChipSeq with various partners ranging from the JGI, Pacific Northwest National Labs, and some people in the UW Biotech Center here. And I wish I could tell you the results of those experiments. We're very eager to see what happens when, in all of this sort of systems biology characterization, when we're actually making ethanol in these strains. But those experiments are ongoing right now, and probably in another 6 to 8 months we'll have results, and we can really tell you something about what the state of the cell is in these fermentation experiments. I should say that in these first generation experiments, we're providing the enzymes exogenously. We're not using yet bugs that are producing them as part of their natural biosynthetic pathways. But what I can tell you about is a little bit of what we've learned from using some of these systems biology approaches to understand anaerobic gene regulation and illustrate some of the approaches we've been taking and some of the directions it's led us in thinking about how we really need to engineer the chromosomes of bacteria to get efficient gene expression in anaerobic environments. And I'm just going to tell you sort of the bottom line from that first, and then tell you a little bit about the technique. So anaerobic gene control in E. coli is regulated by 3 master regulators called ARC-A, FNR, and ISCR. And actually, it turns out one of the most important of these is ISCR. It was discovered fairly recently. These are the various sequences they bind to regulate genes. 2 of them actually don't have crystal structures yet, somewhat surprisingly. But nonetheless, we've been able to use systems biology approaches to sort of tease apart what the regulatory networks are in the anaerobic switch in E. coli. And so these 3 master regulators control sets of genes in programs that look something like this. ISCR actually turns out to be, if anything, the sort of master regulator of this set of anaerobic regulator proteins. It works both aerobically and anaerobically. Aerobically, it controls the metabolism of iron sulfur proteins, and then anaerobically, it collaborates with FNR and ARC-A to coordinate regulation of a set of genes that are required for the pathway switch I just described to you earlier. And there seems to be a hierarchy here where ARC-A is really principally responsible for controlling genes that are involved in energy metabolism. FNR and ISCR additionally regulate glycolysis and fermentation. And so in the pathway that I showed you earlier, if we were to put these regulators in here, ARC-A is sort of controlling the TCA cycle and many of these electron acceptors here. ISCR and FNR are additionally controlling glycolysis and the fermentative pathway here. And so to understand how we put this network together, we've used both gene expression arrays and actually the sort of Chichip and ChipSeq methods that you heard about a little bit earlier today from Rick Young, initially using formaldehyde cross-linking and antibody immunoprecipitation to locate where regulators are on genomes, using first arrays and actually largely converting over now to doing ChipSeq with the JGI to locate where these regulators are bound. And I'll show you just an example of the data that comes from this, actually focusing on this pyruvate formate lyase locus in the E. coli chromosome, looking at actually both Chichip and ChipSeq data here for the RNA polymerase and sigma distribution, either in the absence of oxygen or the presence of oxygen. And this is actually the trace done by ChipSeq here in the presence of oxygen. And you can see an example here where we've picked out the regulatory binding site for FNR, which controls the pyruvate formate lyase gene. Additionally, we've been interested in trying to map the promoters in these regions, particularly because we're interested in trying to use them for engineering anaerobic gene expression. And to do that, we've been using a method, developing a method of high-throughput transcription start site mapping for E. coli. This is work that Kevin Myers has actually been doing, a graduate student in Tricia Kiley's lab. And using an approach of isolating total RNA and then enriching for the triphosphate mRNA populations, removing the ribosomal RNA from it, and then converting that over through a series of steps to something that has Celexa adapters ligated on both ends, and then using Celexa sequencing to get maps of where the transcription start sites are. And so for pyruvate formate lyase, that actually turns out to be surprisingly complicated. There were known to be about 8 promoters, and in this work we found 4 more. So there's actually 12 different transcription start sites here of varying strengths, and actually different changes in regulation and the presence of oxygen, most going up, some actually going down. Pyruvate formate lyase is also controlled by an allosteric regulator that's part of this operon. And when you put this whole thing together, these promoters and attenuator sequences that are in here give rise to about at least 36 possible transcription units that could be controlling the expression of pyruvate formate lyase. So this isn't actually the ideal candidate for thinking about how we're going to anaerobically regulate something. And what we've done is go looking for other locations in the chromosome where we might be able to get a better handle on genes that weren't quite so complicated but gave very good anaerobic control as targets that we could use, for instance, putting in the pyruvate decarboxylase locus. And so to do that, we've used ChipChip to look at the nucleoid organization of E. coli. And I won't go into this in great detail, but the nucleoid in E. coli is organized by 2 proteins that are antagonists of each other, HU and HNS. HNS forms the scaffold. HU helps form the transcriptionally active loops. We've been able to ask, does this genome organization change during anaerobiosis using ChipSeq approaches? I'm going to really map out these antagonistic relationships between where HNS is forming these transcriptionally silenced scaffold regions and where HU is forming these transcriptionally active loops, and this is the distribution of RNA polymerase within them. And then looking in the presence and absence of oxygen, we've been computationally able to figure out that this scaffold really doesn't change much at all when we go from aerobic to anaerobic growth. This just maps out where these regions are. But our attention was drawn in particular to one region where we could see a significant induction of genes anaerobically, and actually zeroing in on those genes, a transcription start site here that really went up dramatically in what was obviously a transcriptionally active loop. It is in between 2 HNS regions here, a gene that turns out to be dispensable, YDFZ. And so this became then a very attractive target for us to think about using to drive gene expression anaerobically. And so we've now focused in on the YDFZ gene, just telling you that from these multi-omic data, we know the start site goes up about 30-fold, the RNA is going up about 40-fold, this dispensable protein goes up about 20-fold. And so this then becomes a good candidate that we can use to fuse pyruvate decarboxylase and get anaerobic induction for executing the strategy I described to you earlier. So I'm going to stop at that point and just, again, tell you these are the people who did the work. And again, thanks for the chance to describe all this to you.